A lot of autism-related genes are pretty ancient, evolving in some of the earliest vertebrates, aka fish, during the Cambrian period. But how exactly did that happen? My name is Dr. Emily Casanova, and I'm an assistant professor of neuroscience at Loyola University, New Orleans, and I study autism, both from the paleogenomics as well as biomedical perspectives. I do research and I teach full-time, building and teaching courses like brain evolution and clinical neuroscience. And if you didn't already know from my last video, I'm also on the autism spectrum. Now, my previous video went unexpectedly viral, so thank you so much from the bottom of my heart. I'm really chuffed to know that there are tons of other neurodivergent folks out there who nerd out on like all the same stuff I do. So thank you. If you haven't watched the earlier video yet, you can find it pinned to my profile, so I do recommend watching that one first and then coming back to this one. Anyways, after the last video, there's been a lot of interest in follow-up, so I thought I'd explain to you exactly how so many of these autism susceptibility genes suddenly evolved in early fish in such a short span of time. Now, back in 2008, Putnam and colleagues published a fascinating paper in the journal Nature where they compared the genomes of various fish to that of the invertebrate animal known as an amphioxus or a lancelet. Now, lancets live in the ocean, and they're tiny, filter-feeding, fishy/worm-like creatures that lie just outside of the subphylum of vertebrates, of us. Now, vertebrates includes anything with a backbone or with a skull, so that includes all fish, amphibians, reptiles, birds, and mammals. Anything else is not a vertebrate. But our little lancelet friend doesn't have any of those bony or cartilaginous features either, so nor do they actually have a true brain, like I talked about in the last video. They do, however, have a centralized nervous system, so not all nervous system related genes are necessarily brain related genes. That's important. The research team in the Nature paper found that when comparing the lancelet genome to that of lampreys, which are a type of jawless fish, as well as to elephant sharks, it became apparent that early fish had experienced 2 rounds of whole genome duplication. Specifically, lancelets didn't show any evidence of this duplication while the other fish did, indicating that these duplications happened after the lineage leading to fish diverged from other chordates like the lancelet. Now, for those of you who know your invertebrate biology, and I'm coming to find that that's quite a few of you, these duplications also were not present in other chordates like the tunicates, which are much more closely related to us. Now, what is a whole genome duplication, you may ask? It's actually exactly what it sounds like. And it's potentially the result of a variety of different things going wrong during cell division in either the sex cells, the sperm or the egg, or very early in the life of the new organism, including errors in the division process, or it can even occur when 2 different species hybridize, which can lead to some problems divvying up the chromosomes. So let's say we have an imaginary creature that would normally have 15 chromosome pairs in the first, but in the first round of cell division of the fertilized egg, the chromosomes are duplicated as normal, but the cell fails to split in 2. That gives the single cell double the number of chromosomes. This fledgling creature now has 30 chromosome pairs with 4 copies of each rather than just 2. Now let's pretend cell division fails again and genome duplication happens once more. Our little creature cell now has 8 copies of each chromosome. Now it sounds bizarre, but whole genome duplications have been known to happen off and on throughout evolutionary history, and provided they don't kill the organisms that have them, which can definitely sometimes happen, they sometimes manage to sweep through a population and come to fixation, so they're basically shared, usually because that population is quite small and maybe somewhat isolated. We see ancient evidence of different whole genome duplications in modern genomes of creatures like yeast, early fish, which we're talking about right now, a group of ray fin fish known as the teleost fishes. That happened later after our own fishy lineage split with them, so we don't share that additional whole genome duplication. The African clawed frog, salmon, and many plant species. So back to our imaginary creature. Now that it has this massive amount of extra genetic material following 2 whole genome duplications and hasn't died, what does its lineage do with all this extra genetic material, and what in the world does it have to do with autism? Now, one of the interesting and predictable things that we see following whole genome duplication over and over and over again is that extra copies of certain gene groups get saved while others eventually get deleted because they don't really matter so much. Now, would it surprise you to know that a lot of developmental genes, including those implicated in autism, are among the genes whose extra copies are preferentially saved after whole genome duplication? That's partly what I studied in my 2023 paper that I talked about in the first video. A lot of autism genes are basically originally duplicate copies of other genes that have been slowly tweaked over time to do new stuff. Now, I'm not going to go into the nitty-gritty of how and why these genes tend to preferentially get saved. Believe me, there's a whole large body of literature that explores this. But if you're interested, I can probably talk about that in a part 3, since it probably needs a video of its own at this rate, because this video is way too long. But suffice it to say that evolution likes to tweak these extra copies of developmental genes over time to do slightly new and cool stuff, which has likely led to the evolution of the vertebrate 3-part brain. More importantly, for our purposes here, a lot of those extra copies of developmental genes that the genome hung on to are in fact autism susceptibility genes. That's how so many autism genes essentially arose all at once. It literally just took the length of time for a couple of rounds of cell division. So, thanks again for all of your interest in my work. If you're interested in reading my original article, the 2023 article, since it's unfortunately not open access, you can find my work email in the bio section under my link tree in my profile, and I'm more than happy to send you a copy. Just pop me an email. Please also let me know if you'd be interested in a part 3. So, thank you and ciao.